Good afternoon, everyone on the East Coast, and good morning to those in the West Coast. My name is Pana Sharma. I'm the CEO and president of Lantern Pharma. As many of you know, recently we created a company called Starlight Therapeutics, and I've been elected as the chairman of Starlight Therapeutics, which is a very, very exciting company focused on a key problem in cancer, and that's pediatric brain cancers. Pediatric and adult brain cancers. So Starlight is entirely focused, Starlight Therapeutics, on CNS, central nervous system, and brain cancers. And this innovation really came to us a couple of years after I joined Lantern. We found that one of our molecules was very, very sensitive in a wide range of brain cancers. And so this gave us some thought and give us some things to consider. And as we develop Lantern Pharma, we continue to increase the capability of our AI platform, more data, more multi-omic data, more pathway models, more mechanistic insights on various types of molecules. And as we came back to one of our drugs, LP-184, drug candidates, I should say, we found that it was very active in central nervous system tumors, and initially observed in GBM, adult GBM. And at the time, we weren't actually even sure that it made sense. And to even exasperate the situation just a little bit, we actually ignored that LP-184 was very active in GBM. Why was that, you ask? Well, it wasn't because of the market, it wasn't because of the indication, but it was really for a more fundamental reason. We just weren't even sure that the drug crossed the blood-brain barrier because we were focused on other solid tumors. And there had been a history of this type of molecule being active in certain solid tumors, most of which, as we developed the molecular characteristics and did more in vitro and then in vivo experiments, we sidelined and really focused on the ones we thought would have real need. But CNS was never lost. And so as we continued the development, we ingested more data, we did more wet lab work. It came out that CNS was not going away. In fact, when we looked at which indications the AI predicted would be most sensitive, GBM continued to be amongst the top 5 or 6 indications, and even with very limited data. So we thought, you know what we should do? We should really validate whether this insight has merit. And we embarked on doing it the way we could at the time, which was in silico, through the use of AI. We decided now to go just from RNA transcriptomic data to large multi-omic data, epigenetic data, methylome data, mutation data. And we were doing this across a wide range of tumors. And we did a campaign. We run our ingesting of data through campaigns. And we had a very large ingest cycle of over 1 billion plus data points, all focused on epigenetic and methylation data across brain tumors. And that to us was a really good telling point, because from there we got signals across a pretty interesting range of adult tumors, brain cancers, adult brain cancers, but also pediatric brain cancers. And that was the aha moment, like, there's something going on here. And we found some correlations between chromatin modeling and also between the levels of PTGR1. And we decided to go ahead and validate that this drug did cross the blood-brain barrier. And so, of course, we did it the way an AI company would, which is using data. We did it through an algorithm, and we weren't 100% happy with it at the time. So we decided, let's do it again using other algorithms. And then as we continued to grow, we said, you know what, let's do it again. Let's look at all the algorithms. And then we looked at the algorithms that were available at the Therapeutic Data Common and decided we can create one better. And so we had a great and brilliant team, and they made their own algorithms to predict the ability of not just our molecule, any molecule. And we tested this across tens of thousands of molecules and eventually hundreds of thousands of molecules, and now we're doing it in millions. And we found actually that we created some of the world's best, highest performing algorithms to to predict any small molecule's ability to cross or not cross the blood-brain barrier, and also give us insights as to its availability. And at the same time, in parallel, we were doing this in the wet lab as well. We ran a number of different assays. We introduced it into orthotopic models of mice and other small animals, and the drug crossed the blood-brain barrier. So in parallel, we found this in silico, we found it in the lab, we found that it was active in a wide range of very interesting tumors, many of which had big, high unmet needs, and in many cases, no cure at all, no real standard of care. And so it became really, really evident to us that we were sitting on a drug candidate that could be a pillar in central nervous system cancers. And so we decided to create a subsidiary of Starlight Therapeutics, which we announced last year. And this year, we plan on launching trials with it. We'll be doing a separate team, a separate management team, and a separate round of funding for Starlight Therapeutics, which is very exciting because it's going after indications both in adult brain and CNS cancers and pediatric, many of which we actually already have orphan indication and rare pediatric designation for. We've applied for several additional rare pediatric and orphan indications. So now we are sitting on a new company that's really literally born from AI. It's born from the data and analytics to understand and to characterize a molecule well before it got to humans. And that's the promise of AI. And actually now we're in humans. We're in a human trial with the molecule as LP-184. And once we get to a maximum tolerated dose, we'll then go into GBM and other adult brain cancers. Once we get to the right point in that development, we'll then take it into pediatric brain cancers, where we're finding some of the highest sensitivity, actually. We're talking single and double-digit nanomolar sensitivity in some of these SMARCB1 mutant cancers and cancers that have what's called chromatin modeling deficiencies. So it's a very exciting molecule and something that we think could be a pillar of modern neuro-oncology treatment.
Thank you, Panna. That is so exciting. And I would like to know more about why Starlight Therapeutic is so unique in its own way.
Yeah, so that's a great question. One, there's not many companies that are just entirely focused on brain cancer. I mean, the market is pretty sizable, $5 to $10 billion globally. It's growing. We're finding new types of brain cancer. The genetics or the biomarker basis of the disease and the knowledge of that is growing quite rapidly. And we felt that there is a real need for a company that's fully dedicated to this growing and pretty significant market. And with the exception of some interesting new agents, one by Chimerix and one by Day One Pharmaceuticals, there's really not a lot of great opportunities to solve for aspects of this disease. And the mainstay still continues to be temozolomide and radiation therapy. I mean, things that have been around for 30, 40 some years. So there's a huge opportunity to create molecules specifically for a range of central nervous system and brain cancers. And the really unique thing about Starlight is we're going into this not hoping that it'll work. We already have significant evidence of its bioavailability of the drug. We have significant evidence of how the drug works in combination with other FDA-approved agents to actually influence different brain cancers. And we're going into this understanding a lot about the mechanistic biology that's going on. And that can help us de-risk and point the drug in the right direction. And so these are all things that typically companies have not done. And most importantly, they haven't done it with an agent that's been exclusively focused in brain cancer. And that's what STAR-001 will try to accomplish.
Thank you. Can you share with us more about where is Starlight at right now? How is it coming to life?
So right now, we have a wonderful leader in Mark Chamberlain, who is our chief medical officer. He did a webinar not too long ago. I urge you guys to take a look at it, where he talks in more detail about the science behind STAR 001 and the design of the trials. We have a trial, a 1A trial that we hope to have a few GBM patients in, and have some signals of safety. And then we're in the process of getting our 1B/2 phase trial that'll be focused in GBM. And we're in the process of getting that okayed by the FDA and getting that ready to launch later this year. We also have sites, some great sites, both community, but most importantly also academic sites, with collaborators lined up for that trial. And we've got a wonderful advisory board that we'll be announcing that has some of the top experts, but very importantly, KOLs, people who will help influence and guide the development of this molecule. So we've got, I would say, a lot of things in order for this thing to be launched very successfully. And we will be doing a separate fundraising. Of course, the shareholders of Lantern Pharma own 100% of Starlight today. It's a 100% subsidiary. But to make this go faster and further, and to really put the focus of value on this asset, we're going to do a separate fundraising for it as its own entity, as Starlight, which will allow us to accelerate the development of this exciting company.
Okay, thank you, Pana. I think that was really great. And I think we will open up the floor for questions. So feel free to send a question through chat, and you can also raise hand so that you can talk to Pana. Okay, I see a question here. One person's asking about the relationship between LP-184 and STAR-001. What are the relationship between them?
Yeah, so LP-184 will continue development as an agent, both mono and combination therapy in non-CNS cancers. But STAR-001, which is essentially the same molecule, STAR-001 is only going to be used in brain and CNS cancers. But essentially, it's the same molecule. It's just given rights to only be developed in brain and central nervous system cancers.
Can you tell us a little more about STAR-001 and its science in brain cancers and GBM?
Sure. Mark Chamberlain did a great job of this in our last webinar, but STAR-001 seems to work really well under two conditions, and both these conditions are very highly indexed in GBM, and so I'll walk through some of these. Number one, when PTGR level 1s are high, the molecule then goes from its prodrug status as a very site-specific, cancer cell-specific alkylator to a drug that then causes double-stranded breaks. So unlike other alkylators that cause single-stranded DNA breaks, this causes double-stranded breaks, and doesn't really give the cancer cell any chance to recover. And so it's got, nanomolar IC50 values, and in some case, even single and double digit in some brain cancers. So it's super potent. We know the drug seems to be more available to the cancer cells than temozolomide, which is kind of the standard of care. And so if you benchmark the bioavailability of this drug with other drugs, temozolomide, other alkylators, it seems to be about twice as more bioavailable. Now, we'll see more as we get further into human clinical trials. Now, the other thing that we're seeing is the intratumoral availability is half. So that to us is a good indication, meaning that the drug is really getting to the cancer cells, and also the potential for side effects because the intratumoral availability is a lot lower, half that of temozolomide. So in terms of the drug crossing the blood-brain barrier, it does it. It doesn't damage neuronal cells. It seems to get the cancer cell. It seems to be double the bioavailability. It doesn't sit around the intratumoral tissue. Its half-life seems to be under 30, maybe 15 minutes that we're seeing. So the mechanics of the drug seem really, really well suited for brain cancer and for a wide range. Now, I mentioned PTGR1. The other aspect the drug really likes is it seems to just come alive when there's any kind of DNA deficiency, DNA repair deficiency specifically. And there can be several types, but the most common are either homologous repair or nucleotide excision repair. Those are the two pathways that are often used to fix the DNA damage. But this drug doesn't really seem to care. So that's really great. Whether it's HRD or NERD, when that's active, this drug is super, super potent. Now, looking at the pathway analysis, and this is how we actually discovered ATRT, we find that if there's chromatin modeling deficiencies, which are related to DNA damage repair deficiency, such as SMARCB1 or perhaps even other HDAC or EZH2, when we find any deficiency there, the drug is also very active. And that's why we are looking at this in ATRT and perhaps also some other diffuse midline gliomas as well. And many childhood and brain cancers have epigenetic deficiencies or chromatin modeling deficiencies. So that's a little bit more about the drug. And the third thing is it seems to work independent of MGMT status. And this is particularly useful because the standard of care today is temozolomide. And, we can look at different papers, but, low-end temozolomide works maybe in 40%, high-end maybe 60%. But either way, about half to more than half of brain cancers don't respond to temozolomide. And the vast majority eventually become resistant to temozolomide. And what happens there is that then there's very few options. And our drug, STAR-001, seems to not care. It works just as well in lowly methylated MGMT as well as highly methylated. So it seems to be independent of this mechanism that GBM seems to use to become resistant to drugs. So we're very excited. It gets to the site where it needs to get to. It's very active. It's got great IC50 values, and it works independent of MGMT status to overcome issues around drug resistance. So that's a great question. Very detailed answer. Sorry for the length, but these are all important subpoints. Any other questions? We'll take one more question.
For a pediatric brain tumor like DMG, we know a single agent is not working. Are all planned clinical trials single agents, or can it be part of a platform trial like PNOC022?
Yeah, we do think that multi-agent for many of these more aggressive pediatric will probably be a good route to take. In fact, for STAR-001, we do expect to have this in combination with spironolactone, which we found seems to make the drug even more sensitive. We also have done a number of combination studies, both with IO agents as well as with other chemotherapies and also with radiation therapy to show that actually increases the efficacy. But great question. Yes, we'll look at monotherapy just because it gives us a clear idea of how the drug works, but it's very high likelihood and high outcome is going to be that it will be used with other agents in practice. So wonderful question. Well, I'm getting the time's up indication from my colleagues. So either I've bored her sufficiently, which is high likelihood, or she has other work to do, which is probably even a more high likelihood. So thank you guys all for joining me on this webinar Wednesday. Thank our team a ton for making this happen. It's not easy. I know in the middle of their workday to kind of drop everything and make these things happen. This was our first one that's live. So if you have feedback, please send it to us. We'd love any of your feedback. And if you have ideas on which webinar Wednesday you'd like to see in terms of topics, please let us know. But again, Starlight Therapeutics, very important, this company would not have been created and born if it weren't for data and AI. And that's where we are today in the world of biopharma development, is that AI is giving us the capability to create whole new companies and whole new indications at a fraction of the time and cost than before. So we're very excited about that. And thank you for joining us. Thank you.